Okay, so we're going to start chapter 16 now, and chapter 16 is on cell communication. So, in chapter 16, we'll be covering the general principles of cell signaling, and then when we meet again in class on Tuesday, we'll go through G protein-coupled receptors as well as enzyme-coupled receptors. Now, the point of chapter 16 is that cells need to sense and respond to their environment. It doesn't matter if it's a unicellular organism or a multicellular organism. So how they sense and how they respond to their environment is really what cell signaling is. So we can consider a simple case, and a simple case would involve a single-celled organism. And the organism we're going to look at is yeast. Normally, budding yeast, which is Saccharomyces cerevisiae, are spherical, and that's what you can see in figure A. However, when they are exposed to an extracellular signal that's known as mating factor, and mating factor is produced by a neighboring yeast, what happens is that the yeast that senses this factor will extend a protrusion towards the source of the factor. So that's what we're seeing in figure B. Now when yeast change their shape like this, so that they have this little extension, they're called a shmoo. S-H-M-O-O. Now what can cause them to change this shape? Well, it's that mating factor, like I said again. And mating factor is simply a protein. And when a yeast senses mating factor, and it's from the opposite sex, it will stop its cell division and reach out towards the yeast that emitted the signal. That's what's happening. So this is a form of cell communication. One cell sent out a signal, and another cell responded to it. Now, when we're talking about cellular information, it can come in many different forms. One form is referred to as signal transduction. And signal transduction is the conversion of an impulse or a stimulus from a physical or chemical form to another form. So I think an easy way to think about signal transduction is to start with a non-biological example. And in this example, we're going to consider a mobile phone. So here's the phone in this figure, although I don't think anybody has phones that look like this anymore. But here we have a mobile phone converting a radio signal. So this is the signal that goes in. And it's going to convert that signal into a sound signal. So it receives the radio signal and then transmits it as sound. Now it's similar in cells. And cells are going to respond to an extracellular signal, and then they're going to transduce that signal or convert it into a different signal on the inside of the cell. So let's look at an example of this. So we can see our signal in, which is this red dot. This is our extracellular signal. It could be a hormone, it could be a protein. It binds to a receptor on this cell. And what's going to happen is now that signal is going to be transduced across the plasma membrane. So it's going to be changed into a different signal on the inside of the cell. So this is our intracellular signaling molecule B. It's different than our extracellular signaling molecule A. This molecule never went inside the cell. Instead, this molecule was transduced into a different type of signal on the inside of the cell. Now what will happen with this intracellular signaling molecule is that it's going to somehow alter the target cell's behavior. And the target cell is this cell right here that received the extracellular signal. Now, we talked a little bit about a single cellular organism with yeast. Now let's talk about multicellular organisms. And multicellular organisms can send out signals in different ways. So we're going to go through a few types of ways on this slide. The most global type of signaling is called endocrine signaling. And this is going to occur, sorry, I'm just going to brighten up my computer. This is going to this is going to occur when endocrine cells release hormones into the bloodstream. And the key with endocrine signaling is that it's going to affect cells that are very far away from the signaling cell. So that's what we can see in figure A. So here's our endocrine cell. It's releasing hormone into the bloodstream, And this hormone is going to travel through the bloodstream and affect cells very far away. That's endocrine signaling. Another type of signaling is called paracrine signaling. And in paracrine signaling, what's going to happen is that a target cell or a signal is going to be released from a cell, and it's going to affect cells that are nearby or in the local vicinity. So paracrine signaling is more local than endocrine signaling. Also, in paracrine signaling, the signal molecules do not enter the bloodstream, and instead they're just going to diffuse locally, staying near the cell that originally secreted them. The third type of signaling we'll go over is called neuronal signaling. And neuronal signaling can deliver messages over long distances, though it is quickly and specifically delivered to individual target cells. And this is because the neuron's going to send that signal through its axon of a nerve cell. Axons can be very long. Now, the fourth type of signaling is referred to as contact-dependent signaling. I like the term juxtacrine signaling better than contact-dependent, but we can use them interchangeably. Now, juxtacrine or contact-dependent signaling is going to occur when two cells bind to each other. So the ligand or the signaling molecule is going to be physically bound to one cell, and it's going to bind to a receptor on its target cell. So you can see that these cells are now making a contact together or undergoing juxtacrine signaling. Now, a fifth type of signaling you should be aware of, though the book doesn't go through it, is autocrine signaling. And this is when the cell that makes the initial signal and secretes it then has a receptor for that signal. So the cell is just signaling to itself. We're going to go through an example of juxtacrine or contact-dependent signaling now. So juxtacrine signaling is very important during embryonic development, and one developmental process in particular is that of the fruit fly nervous system. So when we look at this figure, what we can see is we start off with unspecified epithelial cells. But what's going to happen is that some of these cells are going to become specified. And when they become specified, they're going to become a neuron or a nerve cell. So this middle cell is going to receive signals that tell it to change into a nerve cell. Once it receives those signals, it's going to start expressing a membrane-bound protein that's called delta. So these little red sticks with the ball at the end are called delta, and again, these are bound to the membrane of this middle cell. Now, delta is a ligand, and it will bind to its receptor protein, which is called Notch. Now, we can find Notch on the cells that are surrounding this neuron. Now, it turns out that whenever delta and Notch interact in this scenario, this is going to inhibit these epithelial cells and tell them not to become neurons. So they're going to remain epithelial cells, and then this cell is the only cell that will become a neuron. So this is again an example of juxtacrine signaling. So if you've ever been to a really crowded bar or a really busy party and have tried to talk to anybody there, you know that it can be really hard to listen to that person and filter out all the background noise from everybody else. So what you really have to do is focus on your conversation, filter out everybody else's conversation. Cells really experience a similar problem. So in the same way, they are bombarded with hundreds of different potential signals. However, they must respond selectively by disregarding some of those signals and responding to other of those signals. Now, if a cell is to respond to any signal, it has to have the correct receptor protein in which to do so. Each receptor protein is usually activated by only one signal molecule. So even though a cell may be exposed to 100 signals, if it doesn't have the correct receptor for any of those signals, it cannot respond to any of them. And the way that cells can control what signals they hear or what signals they respond to is by expressing only a limited set of receptor proteins. Now, keep in mind they have the genes to respond, or the genes for all receptor proteins, but that doesn't mean that they're going to express them. And this is what's going to limit the cells and limit what signals they can and will respond to. Now, what's interesting is that the same signal can induce different responses in different cell types. And that's what they're showing you on this figure. So here our signal molecule is acetylcholine. Acetylcholine is a neurotransmitter. Now, when a heart cell receives this signal, what's going to happen is the rate and the force of the contractions are going to decrease. So that's what we see here. So here's our heart muscle cell. Here's the acetylcholine binding to the receptor. This short muscle cell is showing you that it's contracted. So the heart rate is supposed to be higher in this picture. And then when acetylcholine binds, it causes the heart rate to decrease. And that's what's represented by this muscle not being contracted anymore. Now, excuse me, acetylcholine can bind to the same receptor in a salivary gland cell. But instead of telling the salivary gland cell to relax its contraction state, instead it's going to tell the salivary gland cell to secrete its secretory vesicles. Now in C, it's a little bit different because we have the same signaling molecule, which is acetylcholine. Now it's binding to a different receptor on skeletal muscle cells. And when it binds to this receptor on skeletal muscle cells, it tells these cells to contract or to shorten. This is the opposite of what we saw for the cardiac muscle cells. And here's what acetylcholine looks like. Okay, so we just talked about how cells only express select receptor proteins. Now, the receptor proteins they do express can be present in large numbers, and that makes these cells very sensitive to extremely small amounts of the signal. And cells can also alter the amount of a certain receptor that they are expressing, and that makes the cells responsive to a variety of signals. Now, through this combinatorial approach, subtle and complex control is obtained over cell behavior. So that means that two signals may cause a different response than the cell exposed to just one signal or to other signals. And that's what they're trying to show you in this figure. So if we consider the top figure, this cell is being exposed to signals A, B, and C. These are survival factors, so as long as a cell is exposed to these, and it has the receptors to respond to these signals, this cell is going to survive. Now, if the same cell is exposed to factors A, B, and C, and then two new factors called D and E, now this is going to tell the cell to grow and divide. Now, if there's two other signals, and say now we're not exposed to D and E, but instead we're exposed to A, B, C, F, and G, this is a different combination of signals. It tells the cell to differentiate and become a different type of cell. And if we don't receive any of our signals, receiving no signals can also be a signal. And this is, in this case, the signal is for the cell to undergo apoptosis and die. Now, what I want to point out is that there's not just any one given signal that causes a cell to change its behavior. It's usually multiple signals that are going to be necessary for a cell to be able to grow and divide, to differentiate, or even just to survive. Okay, so we talked about how cells are receiving this signal. Let's talk more about the transduction process and how that transduction process is actually going to modulate the cell behavior. So here's our extracellular signaling molecule. Here's our target cell. What's going to happen is this signal is going to bind to a receptor on the plasma membrane of this target cell, and this signal is going to be transduced. So we have an activated receptor. It's going to activate other things in the cell, And this can lead to one of two pathways being activated, or both of these pathways. So the first pathway is referred to as the rapid response, and it's rapid because it affects molecules already present in the cell. So we're going to say that we have some proteins that are already present, but maybe they're inactive. And when we transduce this signal, maybe now we phosphorylate those proteins, or maybe now we cleave an inactivating portion of that protein off. Now, this is a rapid response because the molecule's already made and already present. That protein's inside the cell already. It's maybe not active, but it's definitely there. And once it is activated, now we can alter our cytoplasmic machinery. In this, the example in this slide, and that's going to let us have a altered cell behavior. Now, in the slow response, this is going to cause a change in gene expression. So what has to happen is that our signal has to be relayed to the nucleus. We're going to activate transcription of a gene. It's going to be made into RNA. The RNA will be processed. It will be exported to the cytoplasm. It will be made into protein, and then we now have the protein made that can alter our cytoplasmic machinery, and this is what can alter our cell behavior. Now, this is slow. It takes time to move something to the nucleus, and then to transcribe a gene, and then to process the RNA, and then to move it out of the nucleus, and then to translate that protein. And what if that protein has to go through the ER and the Golgi to be modified? That's why this is called the slow response, is because it's going to take more time. Now, so far, we've just talked about signals and their protein receptors being on the plasma membrane. And I think when we think about a receptor protein, that's where we picture it. And many receptor proteins are indeed found on the plasma membrane. So here in figure A, they're showing you cell surface receptors. And then here's our signaling molecule. Now, some receptors are located inside the cell. And some are not membrane receptors at all, but are instead our enzymes. So that's what they're showing you in figure B. So the receptor here is an intracellular receptor. And not only is it inside the cell, but it's inside the nucleus. Now, with these types of receptors, the ligands tend to be small and hydrophobic. And if they're small and hydrophobic, they can diffuse directly through the lipid bilayer of the cell membrane. And they'll diffuse through, enter into the cell, and then bind to their receptor to elicit a response. Now the large hydrophilic signaling molecules, these are the ones that can't diffuse through the plasma membrane. These are the ones that have to bind to a cell surface receptor. So let's go through an example of a small hydrophobic signaling molecule. And an example of this is a steroid hormone, which is called cortisol. So cortisol is a hormone. It is small and hydrophobic, and it can diffuse directly through the plasma membrane. Now, once it has diffused through the plasma membrane, it will bind to a nuclear receptor protein. Now look, this is an example of a receptor protein that is not bound to the plasma membrane, or any membrane. It's not a transmembrane protein at all. Once cortisol binds to this protein, it alters its conformation, and it activates it, allowing it to move into the nucleus. Once it's in the nucleus, it's going to act as a transcription regulator, binding to the regulatory unit of the gene and activating transcription. So here we can see we're binding to the enhancer, activating transcription, and then we're going to make our RNA. Now each hormone that can directly diffuse through the plasma membrane will bind to a different receptor. And each hormone receptor complex is either going to activate or silence a different set of genes. And now, depending on what type of cell we're talking about, these different sets of genes can be different in different cell types. Now, it turns out that hormones are not the only small hydrophobic molecules that cells use to signal between each other. So cells can use gases to signal too. And these gases, when they're used, will directly bind to and activate enzymes in another cell. This is a form of direct and rapid signaling that's going to alter a cell function within a few seconds. Now, an example of cells that use this type of signaling are endothelial cells. Endothelial cells line blood vessels, and that's what they're showing you in this cross section. So here's a blood vessel. A blood vessel is typically surrounded by smooth muscle, depending on what type of blood vessel you're talking about. And then we have our basal lamina, and then we have our endothelial layer of cells. So here's the lumen of the blood vessel. Now, oftentimes, these blood vessels are innervated, which means that there's a nerve that can communicate with them. And so what will happen is, in response to this nerve cell stimulating the blood vessel cell, or the endothelial cells, is that these endothelial cells will release nitric oxide. The nitric oxide, which is NO, will diffuse from the endothelial cell and into this layer of smooth muscle cells. This is an example of paracrine signaling, because it's diffusing away, it's affecting different cells, but it's only going to affect those cells that are in a local vicinity. Okay, so let's go through this process in more detail. The nerve ending is the cell that's going to release the acetylcholine. This is neuronal signaling. The acetylcholine, which is the signaling molecule, is going to bind to the protein receptor on the cell surface of this endothelial cell. This signal is going to be transduced, that's what's shown by these arrows, and it's going to result in this epithelial cell making nitric oxide. Those are the red dots. The nitric oxide is going to be diffused out of this cell, acting as a paracrine signal, and then diffuse across the plasma membrane of the smooth muscle cells. It's going to bind to and regulate proteins in the cytoplasm of this smooth muscle cell. So this is a rapid response, not a slow response, because we're acting on proteins that are already made. And the end effect is going to be to cause this smooth muscle cell to relax. So it turns out that nitric oxide is an extremely important signaling molecule for relaxing blood vessels. And when you relax a blood vessel, you reduce the workload of the heart, and you decrease the muscle's need for oxygen-rich blood. This is also the mechanism that allows local blood vessel dilation that's responsible for penile erections. Alright, so I was pretty vague about what happens when nitric oxide binds to its target protein right here. So we're going to look at this step in more detail. Okay, so one target protein that's activated by nitric oxide is called guanylyl cyclase. That's what's represented by this blue half circle. Now, guanylyl cyclase, once it's activated, again by the binding of nitric oxide, produces cyclic GMP from GTP. Now, what's interesting about this mechanism is that it's been well studied. And I told you that this is going to relax the blood vessel, and it's important to dilate blood vessels for blood flow for a penile erection. So the drug Viagra is known to enhance penile erection, and the way that it does so is that it blocks another enzyme, and this enzyme would be an enzyme that degrades the cyclic AMP. So if we are not degrading the cyclic GMP, and it's kept in the system longer, it's going to keep those blood vessels dilated longer. That means that this nitric oxide signal is going to be prolonged. Okay, so far we've talked a lot about signaling molecules and how they bind to their receptor proteins. That's this first step up here. But the next step, which we haven't talked too much about, is the intracellular signaling pathway, which is right here. Now, we transduce our signal, especially if we're talking about having a large hydrophilic molecule that's going to act as our signaling molecule. This will bind to its receptor protein, And this signal is going to be transduced or changed as it crosses the plasma membrane. Now we have a signal in a new form, this intracellular signal, and this is going to be on the inside of the cell. This new intracellular signaling molecule will then act to activate another signal, which will activate another signal, which will ultimately activate proteins that are called effector proteins. So these blue proteins are the effector proteins. And effector proteins are the proteins that actually will modulate the cell's behavior. So here we see altered metabolism, altered cell shape, or altered gene expression. Anything that can do this is going to be considered an effector protein. And the final, keep in mind that the point of going through a signal transduction cascade is to alter the cell's behavior or the cell's response to its environment. Okay, so I have the official definition of intracellular signaling pathways on this slide. It is the set of proteins and small molecules that interact with each other to relay a signal from the cell membrane to its final destination in the cytoplasm or the nucleus. Now, after the extracellular signal binds to the receptor protein, that signal is transduced into an intracellular signal. Now, this initiates one or more signaling pathways that relay that signal in the cell. So we may activate a protein, which activates another protein. This signal can be transduced again. So if you change the signal's form, that would be another transduction. Or it could be amplified. And when you amplify a signal, you're going to make the signal stronger, such that only a few extracellular signaling molecules are required to evoke a very large intracellular response. So you can see at this step, even though we only had one extracellular signaling molecule, that signal was transduced across the cell membrane. Here's our new signal. Here we transduce that signal again because we're going to end up with a new signal. Now we don't just have one of these new signals, we have multiple ones. That's where the amplification comes from. This signal can then be integrated. So that means that they can change or they can come together before the intracellular signaling pathway is to move forward. So here you're seeing multiple proteins that need to come together in order to integrate this signal. That means that you may have to have multiple pathways that are also activated in order to integrate the signal. And finally, the signal can be distributed to one or more signaling pathways or effector proteins to create a branch in the information flow diagram. So this is how cells can have complex, different responses to the same set of signals. So it really depends on other signaling pathways that are feeding into this original pathway, as well as what effector proteins are going to be altered. I'm going to finish this lecture by talking about molecular switches. So many key intracellular signaling proteins behave as molecular switches. And a molecular switch is a protein that can toggle reversibly between an active and an inactive conformation. So once they're activated, they remain activated. And that means that they're going to stay in their active state, outputting a signal until another process switches them off. So this means that every time a protein is activated in a signal pathway, it has to be deactivated before that pathway can be reset. Both the activation and deactivation are equally important in the signaling process. There are two main classes of molecular switches. The largest class are proteins activated and inactivated by phosphorylation by a protein kinase. So that's what we're looking at in Figure A. So in Figure A, we have our inactive protein. Then we have our signal in. So this means that we had some kind of signal and it was transduced, and it's going to be enough to activate a protein kinase. A protein kinase is going to take ATP, exchange it to ADP, and take that phosphate from the ATP and put it on this protein. Now this protein is considered on. It's activated. And as long as it's activated, it can put the signal out, so it can be activating other proteins. Now it's going to continue to activate other proteins until another protein acts on this protein. And this protein is a phosphatase. Phosphatases remove phosphate groups. Once this phosphate group is removed, this protein is no longer on. It's now considered off, and it will remain off until acted on by a protein kinase. Kinases are proteins that phosphorylate other proteins. Another class of molecular switches are the GTP binding proteins. And these proteins switch between an active and inactive state depending on whether or not they have GTP or GDP bound to them. Now once activated, and again they get activated by GTP binding, these proteins have a GTPase activity, and they will shut themselves off by hydrolyzing their bound GTP to GDP. So that's what we can see here. So here we have our GTP protein on. Now it has its own intrinsic GTP hydrolysis activity. So after a certain amount of time, it's going to undergo GTP hydrolysis, and that's what's going to remove the phosphate and turn the protein off. Now when it needs to be turned on again through some signal in, the GDP is going to be removed and GTP is going to be put back on this protein. So this is a GTP exchange for GDP. This again turns the protein on, and it's on because it's bound to GTP. And as long as it's on, it can be putting the signal out. Now, it'll have an output signal until its own intrinsic GTPase activity or GTP hydrolysis activity removes the phosphate and turns it off. Okay, so that's where I'm going to stop with Chapter 16. We'll cover the rest in class. So again, write down 3 questions that you have on the end of Chapter 15 and the beginning of Chapter 16, and turn it in before class on Tuesday, and you can get 5 extra bonus points that I'll count towards your total exam score at the end. Thank you.